Today we have the great pleasure of having with us Antonio Riva, who is the team leader of liver immunology at the famous Roger Williams Institute of Hepatology, and in King's College of London. Antonio is one of the leaders in the world of liver immunology, and today he will address a very fascinating topic, which immunomodulatory strategies for alcoholic liver disease. Why, how, and when? Antonio.
Thank you, Professor Tiribelli, for the very kind introduction.
My name is Claudio.
Perfect. I will share my screen. Let's see. Can you see my screen?
No.
No, you can't see it.
No, we can see you, but not your screen.
Ah, okay. Let me see.
Now we can see your screen.
Now, fantastic. Okay. So thank you for the kind invitation to speak for this, your very important series of webinars in liver sciences. It's an honor also to be speaking, being Italian, speaking for the Italian Foundation for Liver Disease. It feels like coming back home in a way. I'm very happy to be here. I will talk to you about this very interesting and crucial topic, really. So can we apply immunomodulation in alcohol-related liver disease? Why would we do it? How would we approach this? And when is the right time to do this? This is just one of many studies that we carry out in my immunology and alcohol team here at the Institute of Hepatology. We are actually an independent research organization. We only have academic affiliations with King's College London. And I will take you through a series of introductory slides, and I will also present some of our published and few unpublished data as well. So I'm sure you all know these graphs, so I will spend very little time. This is just to introduce the concept that the burden of liver disease is steeply increasing. And we know that one of the main causes of this increase is alcohol consumption, particularly in some European countries more than others. So there is a direct relationship between the amounts of units drank and the probability of developing liver disease later on in life. This is the way we conventionally describe the progression of alcohol-related liver disease. We know that, first of all, a normal liver develops some steatosis, fat accumulation. This can progress through to fibrosis, and until now, the disease is still reversible. When we hit the moment when fibrosis transforms into cirrhosis, this is when the real problems start happening, because cirrhosis is conventionally considered to be irreversible. And obviously, we have a much higher predisposition to develop hepatocellular carcinoma or to decompensate, which is a phenomenon that increases mortality significantly. On the side of this more conventional path, we also have a different disease entity, which is alcoholic hepatitis. This, as you know, is the most inflammatory type of alcohol-related liver disease. There is a very strong immunological component to alcoholic hepatitis, and it doesn't necessarily have to occur on the background of cirrhosis. But patients who develop alcoholic hepatitis, they are at severe risk of immunological dysfunction, bacterial infection, and higher mortality. We know that at the moment there are no specific treatments for alcohol-related liver disease. Alcohol abstinence is still the main cornerstone of treatment, let's say. Most of the rest involves supportive measures, nutritional measures. Many of these patients, because of the increased risk of bacterial infection, tend to be preventatively treated with antibiotics, broad-spectrum antibiotics. But the problem with this is that this can increase the selection of antimicrobial resistant strains, which is an increasing problem, not just in Europe, but worldwide at the moment. Liver transplantation is mostly not an option for many of these patients, and there is still a lot of stigma associated with the development of alcohol-related conditions. And immunomodulation, particularly with steroids, seems to be effective in a subset of patients, but the pharmacological benefits cease to exist if bacterial infection develops. So we are in a situation where we need new specific treatments, new effective treatments. If we look at the burden of liver disease in relation to the amount of resources that are spent in research for new treatments, it's very clear that alcohol-related liver disease, out of these four main causes of chronic end-stage liver disease, is the one that has the highest burden in terms of hospitalization costs, liver transplants, and enrollment in studies for cirrhosis, fibrosis, and also increased mortality. However, if we look at the amount of resources that are spent towards addressing this disease, alcohol-related liver disease is the one with the least amount of resources spent. So there is a very unfavorable attention to burden ratio that puts ALD really in a very inadequate position. Why is this important to say? Because the mean efficacy of these current non-specific treatments in alcoholic hepatitis, at least, is very low. We know that there is a very strong direct relationship between the amount of research that is performed and the mean efficacy of the drugs that are developed. So we really need to boost research in alcohol-related liver disease. And I hope that what I'm going to talk to you about today could be a new potential, very novel avenue, actually. So what happens to immune responses in alcohol-related liver disease? Well, as we see that the disease progresses all the way through cirrhosis, potentially alcoholic hepatitis, and hopefully not all the way to acute and chronic liver failure, there is a switch in the way the immune system works. On the one hand, we have systemic inflammation that increases progressively, but at the same time, this is accompanied with a suppression of antipathogen specific responses. We know that this is not only bacterial in terms of antibacterial immunity. I mean, we know, for example, that cirrhotic patients tend to have lower responses to vaccination. And this affects, obviously, a completely different set of diseases, not just, bacterial infection, as I said. But because of this switch in immunological functionalities, there is an increased burden of infection, including opportunistic infections, that are really one of the main causes of death in these patients. The immune system works along gradients of balancing factors. On the one hand, we have factors that regulate inflammation or immunopathology. On the other hand, we have factors that boost immunoprotection. And what we believe is happening in patients with alcohol-related liver disease is that we see these the development of a very unbalanced situation where immunopathological mechanisms are increased, and this has an effect of reducing immunoprotection. And in our studies and those of many other groups, we see that this impacts both innate and adaptive antipathogen immunity, but also more barrier-like type of responses, like mucosal immune cells at the intestinal barrier, for example. And in the end, if we consider the anatomical relation that exists between the liver and the intestine, we could see the liver as a sort of semi-mucosal organ. And in fact, there are a lot of innate-like immune populations that reside in the liver, and they really contribute to this firewall activity that the liver performs, you know, placing itself in the interface between the gut and the rest of the body. Immune checkpoints are one of our main key areas of research, and we have published a lot on this. So I will talk to you mostly about these molecules, also because with the revolution of cancer immunotherapy, they have really come to the fore. So very briefly, what are immune checkpoints? Immune checkpoints are a vast array, a vast set of molecules that regulate, that fine-tunes really, really well the activation or the suppression of immune responses. Immune checkpoints are a physiological mechanism that either participates to activate immune responses in the first place, or inhibits immune responses when these are no longer required. For example, when an infection has been cleared and we need the immune system to shut down again in preparation for the next, including generation of immunological memory and so on and so on. In the context of alcohol-related liver disease, we would see activation as inflammation, and we would see inhibition of immunity as immunosuppression. And this is the balance that we believe is altered, and we believe that immune checkpoints play a big role in this. Immunotherapeutic approaches go mostly by blocking this interaction between receptors and ligands, both in a stimulatory synapses and inhibitory immunological synapses. And what our approach, the approach that we want to present, is that probably blocking both pathways in ALD could be a very good way of rebalancing this dysfunctional immunity. There are a lot of different pathways. This figure is from 2017, but I can tell you that as far as we know now, we could quadruplicate the amount of pathways that we know now to be immune checkpoints, and this is increasing weekly. But initially, when all these studies were started, really the focus was on two key pathways, PD-1 and TIM-3. PD-1 has PDL1 as its main ligand. TIM-3 has galectin-9 or SICAM1 as its main ligand. SICAM1 is very relevant for cancer because it's actually a very important cancer antigen. What we discovered first was that during the progression of alcohol-related liver disease, there is an increase in the expression of these strongly inhibitory pathways on circulating immune cells. And this affects non-specifically PD-1, PDL1, so ligand and receptor, but also TIM-3 and galectin-9, again, ligand and receptor. In this study, we managed to correlate this upregulation of suppressive pathways with a loss of antibacterial interferon gamma responses and an increase in immunosuppressive IL-10 based responses, therefore indicating a switch in the ability of the immune system to respond to bacterial insults. But in the same study, we tried to block PD-1 and block TIM-3 to see if we could achieve immune reconstitution. And this was a first in approaching this in alcohol-related liver disease. And we found that indeed by blocking PD-1 and TIM-3, we could suppress immune inhibition and restore a more physiological balance in interferon gamma production, reducing at the same time immunosuppressive interleukin-10. So this was a groundbreaking finding that really prompted us to think and believe that immunomodulation in ALD may be a very valid new approach to apply. The immune checkpoint system is a little bit more complicated, and over the years it has become a little bit more complex when we discovered that immune checkpoints can also exist as soluble molecules that can travel around the body through the systemic circulation, translating these stimulatory or inhibitory activities at a distance. So what does this mean? It means that we don't even have to be inside the target organ where the disease develops, but through the secretion of these soluble molecules, we can affect the immune responses peripherally and systemically. We know that these molecules are biologically active, and these are just a few old papers, but now the literature is really increasing in this respect. And I've just published an editorial last week on the importance of soluble immune checkpoints in human disease in Frontiers in Immunology, because there are still a lot of unanswered questions and we don't know about these molecules. But one thing we know for sure is that they are biologically active and they perform strong immune regulation, immunoregulatory activities, even stronger than pro-inflammatory or anti-inflammatory cytokines. Because of our findings in terms of PD-1 and TIM-3, we refocused our studies on the soluble forms of these molecules to see if the soluble TIM-3 pathway or the soluble PD-1 pathway, I don't know why it doesn't show here, but to see if these soluble pathways were also dysregulated in alcohol-related liver disease and whether we could target them to reconstitute an efficient antibacterial immunity. And this was published, again, a couple of years ago now. So why TIM-3? TIM-3 because it's a very crucial molecule in boosting immune suppression mediated by a lot of other checkpoints. In the cancer immunology field, we see that adaptive anti-cancer cells who express TIM-3 as well as PD-1 or LAG-3 or other checkpoints, these cells are far more suppressed in their functions than those that do not express TIM-3. And TIM-3 has some quite complex pathways of signal transduction intracellularly, but they both, or most of them, end with inducing T-cell suppression, particularly antigen-specific suppression of responses in terms of TCR-mediated signal transduction. The two main ligands of TIM-3, as I said, are CEACAM1 and galectin-9. CEACAM1 is very important because it stabilizes the structure of TIM-3, making it more likely to transduce inhibitory signals. And what is interesting about TIM-3 also is that just outside the transmembrane domain, it has a pretty specific cutting truncation site where disintegrin metalloproteases ADAM10 and 17 can cut the molecule, therefore releasing the full extracellular domain, including the ligand receptor epitopes. So we collected several patients with alcohol-related cirrhosis and severe alcoholic hepatitis, and we did a pretty broad screen to assess the levels of soluble checkpoints, and we quantified 16 in this study. When we ran our group comparisons, we saw that TIM-3 and CD80, unexpectedly, showed up as the most highly expressed, particularly in patients with severe alcoholic hepatitis. TIM-3 in particular was extremely strong at discriminating patients compared to healthy controls. And TIM-3 survived as the main driver of differences even after multivariate deconvolution and decorrelation to try and balance out all the background noise that usually affects the intercorrelation and the redundancy in these immunological systems. I would also like to point out that soluble PD-1 resulted the least different in this case. So even though PD-1 has a very strong effect as a membrane receptor, its soluble form doesn't seem to be associated in any form or shape to progression of alcohol-related liver disease. And also because of these results, we then focused our attention particularly on the soluble TIM3 pathway. When we measured the ligands for soluble TIM3, we also saw the soluble galectin-9 and soluble CEACAM1 are equally upregulated and equally overexpressed in patients, in ALD patients, particularly those with severe alcoholic hepatitis. And what this led us to believe is that it is not just an alteration that affects one of the molecules in this pathway, but is the entire pathway that is completely dysfunctional in the progression of ALD. So to this effect, we decided to block the entire pathway to see if this would achieve immune reconstitution. So we set up an in vitro culture system where we treated peripheral blood mononuclear cells from healthy controls and patients with severe alcoholic hepatitis with lactose, lactose can block galectins and therefore blocks the interaction between galectin-9 and TIM3. And we also provided an excess of soluble TIM3 to try and achieve further blockade of galectin-9 and soluble CEACAM1. And what we found is that by achieving this combined blockade of the entire soluble TIM3 system, we could indeed achieve immune reconstitution in patients with severe alcoholic hepatitis. And this was assessed both in terms of interferon gamma producing cells and in terms of IL-17 producing T cells in an antibacterial fashion. What is also interesting to point out is that we did not overcompensate for this in healthy controls, indicating that this blockade has effect only in subjects where the system is dysfunctional in the first place, which obviously has relevance in terms of safety, because we did not exacerbate hyperimmunity in subjects that were healthy controls. One thing that also furthers this idea is that we could also restore IL-10 production in combination with interferon gamma and IL-17. And this to us means that we have reinstated the balance between immunity, but also control of hyperimmunity. And this is very important. There have been, since our reports, other reports confirming this same data. This paper was published in 2020, and they also indicate the soluble TIM3 is higher. And this paper is a little bit more recent and splitting alcoholic hepatitis patients by MADRS discriminant function. We see the soluble TIM3 tends to be higher in patients that are doing worse. However, how effective or how safe these molecules are to actually use in vivo, we don't know that. We have some indication from the cancer immunotherapy revolution that immunomodulation using antibodies that target immune checkpoints is very effective and is also relatively safe. Obviously, we know that there are some adverse events that occur during immunotherapy treatments, but we also know that most of them are relatively manageable. And the occurrence of severe adverse events is not as prevalent. I also like showing this picture. This is a metastatic melanoma that after one dose of anti-PD-1 and anti-CTLA-4, in 3 weeks disappeared completely. And the patient describes seeing the tumor melting in front of her eyes. So these treatments are extremely powerful. However, one issue with these studies is that cancer itself is not a hyperinflamed condition. And what we don't want really is to further trigger hyperinflammation in ALD patients where hyperinflammation is already driving disease. So an answer to this issue comes from studies that were carried out in bacterial sepsis, which, as we know, is another strongly hyperinflammatory condition. And in many preclinical studies, they all report that there is a survival advantage significant when administering immunotherapy in terms of anti-PD-1 and anti-PD-L1 in these animal models of severe bacterial sepsis. And this immune checkpoint inhibition, or ICI, advantage is also maintained when adjusting for antibiotics or for type of challenge or site of challenge. PD-1 and PD-L1 pathways show to have the predominant advantage, and they also have additional benefits over antibiotic therapy alone. And it's really these preclinical results in animal models that boosted the possibility to translate this into humans, and 2 clinical trials for anti-PD-1 and anti-PD-L1 were then carried out in human patients with bacterial sepsis to assess safety and efficacy, but primarily safety of these treatments in a potentially hyperinflammatory condition. The results of both these trials were extremely powerful in showing that these immunotherapy approaches are potentially effective and safe in hyperinflammatory conditions. There was, in both trials, there was indication of immune reconstitution by 28 days, and this was measured by increased leukocyte count and also HLA-DR expression on monocytes. There was no induction of pro-inflammatory cytokines, and actually IL-6 and IL-8 reduced over time. And this was safe, so there were no unexpected adverse events given the biology of the inhibitors tested. So we see that there is definitely potential for immunotherapy approaches in a condition where hyperinflammation and immunological dysfunction and disequilibrium are key drivers of disease. One thing that we have to always consider is what are the consequences of this immunological imbalance. And the consequences are obviously an increase in mortality associated with microbial infection. But what we also have to consider is what comes before the establishment of this paradoxical immunological profile. And what we believe triggers most of these occurrences is gut alteration of the intestinal system and an increase in bacterial translocation, because we see that this develops really in parallel with the establishment of this immunological, dysfunctional immunological profile. So we always need to go back to one of the main mechanisms also responsible for this increase in bacterial infection, and this is the translocation of commensal bacteria. We know that this is a problem in subjects who drink alcohol, because ethanol itself can damage the gut. Alcohol can damage the intestinal epithelial cell layer, but it also triggers powerful changes in the intestinal microbiota with a shift in the production of beneficial molecules such as short-chain fatty acids. There is a reduction in lactobacilli, and there is a reduction in bacterial species that help control the integrity of the intestinal barrier. Once the intestinal barrier is dysfunctional, the illuminal content in terms of bacteria, bacterial metabolites, toxins, can translocate across into the portal circulation, where it reaches the liver, and through the liver then disseminates systemically. And we see that there is an establishment of this sort of vicious circle where translocation induces inflammation systemically. Inflammation further damages the gut lining, and this obviously perpetuates the cycle. And at an intestinal level, different layers can be rendered dysfunctional. There is alcohol effects on the microbial communities. The mucus layer itself can shrink and can change in chemical composition. the tight junctions, so the intercellular junctions, are damaged, and this favors the passage of bacterial luminal matter into the mucosal site. And from the mucosal site, it then translocates into the venous circulation and so on, completing the circle as I described it. We ran a pretty broad transcriptomic assessment in the alcoholic gut, let's say, using biopsies from patients with alcohol-related cirrhosis. This is still currently unpublished data, but we see that the alcoholic gut is very different compared to the healthy control gut, with a lot of upregulated and downregulated genes. And most of these genes actually end up relating to intercellular junction pathways, where we see that these pathways are strongly upregulated in patients, which is an obvious indication of ongoing damage and ongoing compensatory active repair mechanism. We know that locally in the gut, there is inflammation, and we show this very clearly, very elegantly with this paper that we published in 2020, where we assessed plasma markers of inflammation, but also fecal markers of inflammation. And we quantified the pool of very conventional cytokines in plasma samples and matched fecal samples from patients with stable cirrhosis or acute decompensation. And what we found was that the same cytokines that we could detect in plasma and feces were associated with markers of liver disease, as much as the same markers as are conventionally associated when assessed in the plasma. So what happens in the intestine has a clear reflection in what happens systemically and vice versa. So there is definitely a bidirectional crosstalk between the two systems. Studies in terms of intestinal permeability in ALD are not a new thing. We've been known for quite a few years now that there is increased gut permeability in alcohol-related liver disease. And this was assessed, for example, with PEG, this detected in the urine. We know that this increased gut permeability raises the levels of bacterial markers in the peripheral circulation. In this paper from 1989, this was assessed in terms of bacterial lipopolysaccharide in the peripheral circulation. But we also know that there are increased plasma indicators of exposure to bacterial antigens in these patients. So not only do the bacteria leave the gut, they definitely translocate systemically, and the immune system reacts to this exposure by secreting, in this case, endotoxin receptors to try and neutralize this bacterial effect. What we found in our course of patients matches this entirely, both in terms of lipopolysaccharide, but also in terms of D-lactate. So D-lactate is an enantiomer of lactate, but is only of bacterial origin, because our body, our cells do not produce this D-lactate form, and they do not metabolize it. So the detection of D-lactate in the peripheral circulation is a clear indication that it can only come from some form of bacterial agent. And in our case, we use it as a pretty strong marker of bacterial translocation. We know that endotoxin and D-lactate are strongly directly correlated with indices of increasing liver disease severity in ALD patients overall, but also specifically if we restrict our attention to patients with severe alcoholic hepatitis. So there is a clear link between bacterial translocation, disease severity in these patients. And we also find that this correlates really well with the immunological dysfunctions that we have detected in terms of soluble immunological immunomodulators. This is our friend, soluble TIM3, in correlation with D-lactate, or soluble CD163, which is also a marker of immune exposure to bacteria in the peripheral circulation. And obviously, we see that this really goes very well in parallel with increasing levels of inflammation in these patients, where all the conventional pro-inflammatory cytokines happen to be strongly upregulated in patients with severe alcoholic hepatitis in particular. Now, why is this relevant? Why have I made such a big digression on bacterial translocation? Because bacterial translocation is directly responsible for the hyperexpression of suppressive checkpoints on antibacterial T cells in these patients. We found, in fact, that using a pretty basic dose response titration curve for LPS in vitro, we found that LPS itself can increase the expression of PD-1 and TIM3 on antibacterial T cells in the peripheral circulation. And the reason why we are so certain that this is mediated directly by bacterial interaction is because when we blocked the detection of lipopolysaccharide using anti-TLR4 and anti-CD14, here you see again our dose response curves with increasing PD-1 and increasing TIM3 over a dose response curve of LPS. So when we blocked the detection of LPS with anti-TLR4 and anti-CD14, this completely abrogated this immunosuppressive response, both in CD4 and CD8 cells in an antibacterial fashion, and including both PD-1 and TIM3 pathways. So yes, it goes through monocyte detection of bacterial products because of TLR4 and CD14, but the effect is directly linking bacterial translocation with dysfunction in immune checkpoints. And we also know from a real-life cohort of patients with alcohol use disorder that translocation precedes disease, because if we assess D-lactate in a real-life AUD cohort that is only assessed based on liver stiffness, we see that even in subjects where liver stiffness is very low, so in the range where normally we would find healthy subjects, D-lactate is very high and is actually much higher than what is found in subjects with AUD, alcohol use disorders, with high liver stiffness in a range of values where we would normally cluster fibrosis and particularly cirrhosis, so severe liver disease. This cohort of patients was monitored through their GPs. So these were not patients that were recruited through hospitals. They were not known based on existing diagnosis of liver disease. So that's why we say this is a real life cohort. Now, when we compare this with the range of values that are detected in conventionally recruited patients, we actually see that the levels of D-lactate, so the levels of bacterial translocation that is observed in subjects with low liver stiffness, this range is actually overlapping to what we find in patients with severe alcoholic hepatitis. And these patients, these subjects, well, they are not patients yet. So we believe that translocation is a phenomenon that precedes development of disease and may be one of the main drivers. Interestingly, the levels found in the high liver stiffness group compare really well with those found in the conventionally defined alcohol cirrhotic group, just showing that there is parallelism in patients with advanced disease already established and those that are probably on the way to being assessed as such. And this is just to highlight the main difference that we believe precedes the establishment of disease. So what happens in the gut is that when there is a pathological break of the barrier, there is gut leakiness, and there is a direct contact between bacterial matter and immune cells in the mucosa. and particularly antibacterial mate cells play a big role in this. So mate cells are T cells that respond to bacterial metabolites, not bacterial peptides. So they are very unconventional, but they are the main sentinel system against bacterial infection in the gut and in the liver. What we found some time ago was that when we expose healthy mate cells to bacterial products obtained from stool extracts, we observe a reduction in the amount of mate cells that differentiate as such. They become more activated, non-specifically, but they also reduce their functionality when they are further stimulated with bacterial antigens. Now, these findings come from an in vitro setup, but they match perfectly what is already seen in the peripheral circulation of these patients. So patients, particularly those with severe alcoholic hepatitis, they almost do not have mate cells in their peripheral circulation, but the few that remain are non-specifically hyperactivated, but they are not responding to further bacterial stimulation. And this is directly correlated to alterations that increase bacterial translocation in the same patients. So there is a lot to do with mate cells. They can be reprogrammed, they can be derived from iPSC, we can restore the gut integrity, but one of the main future avenues that is very exciting is the one of microbiome editing, because we know that, for example, some specific bacterial species can change the way mate cells are induced or they are suppressed in the way they function. We can change the balance between the species that produce metabolites or not, but also we can do some metabolic reprogramming, if I can call it like that, or metabolic engineering in antibacterial immune cells by acting on the microbiome. This is a paper that was published earlier last year, and it shows how alcohol impairs immunometabolism in naive T cells. To go straight to the key results, which is the metabolic assessments, these authors show that alcohol shifts the balance in the metabolic energy production in T cells by boosting glycolytic pathways and suppressive oxidative phosphorylation. And also, obviously, because metabolism is very important for immune cell functions, any metabolic shift can have an effect on an efficient antibacterial immunity. And what the authors found was that indeed metabolic dysfunctions were associated with gene expression changes that relate to autophagy, and autophagy is very powerful in modulating the way immune cells work. Now, these changes in gene expression, we also found them in mate cells, where we see almost a complete loss of some transcription factors that are key for their antibacterial functions. And when we went on to investigate a little bit more of transcriptional regulation in patients with liver disease, this is a very small unpublished study from one of my students, we found that the transcriptional regulatory network in the tissue itself is heavily dysfunctional and can discriminate really well patients who have alcohol-associated liver disease from healthy controls. And this study highlighted also some completely novel genetic targets that can be utilized as possible new pharmacological targets or agents, let's say. So just to conclude, why do we want to do immunomodulation in ALD? Well, because we know that this intestinal damage with bacterial translocation is linked to immune alterations. We know that these immune alterations really impact immune regulatory pathways that can be targeted with immunomodulation. particularly checkpoints, we know that these alterations cause a suppression of antibacterial responses, and that this is directly correlated with bacterial translocation. So one possible way to achieve immunomodulation could be blocking bacterial translocation, blocking detection of translocated products, because this would prevent immune exhaustion in these patients. We can also block the immune checkpoint pathways, and we have shown that this can reconstitute a state of efficient antibacterial immunity. And even if we target these pathways in a soluble form, we can still achieve a pretty consistent immunomodulation without triggering unwanted immunological side effects. And this is also validated by some immunomodulation in vivo studies and those 2 clinical trials in sepsis that I talked to you about earlier. There are also a lot of molecular interventions now that we can perform, particularly targeting the microbiota and possibly acting on immunometabolic pathways to restore an immunocompetent state in these patients. So this is really just to talk a little bit about the when, and I'm not going through all of these points really because I have discussed them already, but when do we target? Well, we can target early on. We can target even when the damage is established, because by blocking PD-1 and TIM-3, once exhaustion has set in, we can still reconstitute some form of immunity. And in terms of when is the right time to initiate these strategies, I would say that the right time is now, because we need new efficient treatments and specific treatments for alcohol-related liver disease. Because if you remember from my initial slides, there is a lack of effective treatments, and alcohol-related liver disease is where we have to really invest in developing new resources. Just to conclude, I would like to acknowledge the Roger Williams Institute of Hepatology, where all these studies have taken place. My team, specifically with my research assistant and all the students that over the years have taken part in these studies. These are all the clinical collaborators and scientific collaborators that have helped recruiting patients. These are some of our funders, and obviously I would like to thank you for your attention.